Welcome to Edifysity, where learning is simplified. In previous videos of this module, we have discussed what is the anatomy of kidney, what are the different blood vessels and nerves that supply to kidney, and what are the different parts of nephron, and what are the different types of nephrons which are present in human kidney. Now, in this video, we are going to discuss about histology of the nephron and collecting duct. A single layer of epithelial cells forms the entire wall of the glomerular capsule, renal tubule, and ducts, but they differ in histological features. So glomerular capsule have two layers. First one is parietal layer and second one is visceral layer. Parietal layer is formed by simple squamous epithelium which forms outer wall of the capsule, whereas visceral layer is formed by simple squamous epithelial cells which are modified and they are called as podocytes. Podo means foot, cyte means cell. So these cells have foot shape. From these cells, many foot-like projections arises which are called as pedicels. This visceral layer forms the inner wall of the capsule. So glomerular capsule is best understood with an example of fist punched into a limp balloon. Histoarchitecture of glomerular endothelial cells, basal lamina, slit membrane and podocytes is best understood with this pictorial representation. Glomerular endothelial cells have large pores and are leaky in nature, but they prevent filtration of blood cells, allows all components of blood plasma to pass through. Basal lamina of glomerulus lies between endothelium and podocytes, prevents filtration of larger proteins. Podocytes form pedicels between which are filtration slits. So above the basal lamina, there is slit membrane between pedicels. This slit membrane prevents filtration of medium-sized proteins. So when we start with renal tubule, the first part of renal tubule comes into picture, that is proximal convoluted tubule. Proximal convoluted tubule is made up of simple cuboidal epithelial cells with prominent brush borders of microvilli. These microvilli, like those of small intestine, increase the surface area for reabsorption and secretion. Second part is loop of Henle, that is descending limb and thin ascending limb. It is made up of simple squamous epithelial cells. Third part is thick ascending limb, which is made up of simple cuboidal to low columnar epithelial cells. When we discuss about thick ascending limb, another modified portion of thick ascending limb comes into picture, that is Juxtaglomerularapparatus. This Juxtaglomerularapparatus is formed by two layers. First one is macula densa, second one is juxtaglomerular cells. Macula densa is the final part of ascending limb that is in contact with efferent arteriole. The name densa is given because cells are crowded together in this region. Second type of cells are juxtaglomerular cells. They are modified smooth muscle cells of efferent arteriole which are present alongside of the macula densa. These two cells togetherly they are called as juxtaglomerular apparatus. This apparatus helps to regulate blood pressure within the kidneys. Next part of the nephron is distal convoluted tubule. Most of the distal convoluted tubule is made up of simple cuboidal epithelial cells. Last part of the distal convoluted tubule and all of collecting ducts, they are made up of simple cuboidal epithelium cells consisting of principal cells and intercalated cells. Principal cells have receptors for both antidiuretic hormone and aldosterone. These two hormones regulate their functions. Second type of cells are intercalated cells, less in number, play a role in homeostasis of blood pH. So in this video, we discussed about histology of nephron and collecting ducts. In our next video, we are going to discuss about other parts of urinary system like ureter, urinary bladder and urethra. Thanks for watching.